Hello everyone, and welcome back to another lab video. In truth, this is a reiteration of a previous lab video I did, extracting DNA. And the reason I'm redoing it is because I didn't realize the audio from the previous video that I uploaded had actually cut out at the halfway point. And I finally got a new camera, so the quality on this one is going to be infinitely better. Well, it's more accurate to say I have a new smartphone, but regardless, the camera on this one works. So I actually don't have to record at like 20 or 30-second clips at a time and then compile it all together later on. So I'm going to be rehashing the DNA extraction lab with a few differences this time. Going to be using strawberries instead of bananas, and well, a strawberry rather, and I'm actually going to be describing the science and mechanisms behind the extraction itself before we do the lab. Now, if you've seen this video, or if you've done it in class before, which is incredibly likely because there's like a million of these on YouTube, then you don't have to watch this video. All I'm doing differently is explaining the science behind it. If you want higher production values, there's like a billion YouTube videos that you can watch. So I apologize if this is old hat for some people. But before we do the extraction, we're actually going to have to look at a couple words, vocabulary words, to learn what they mean and how the extraction itself works. So first of all, everybody knows what DNA is. DNA is basically instructions for building protein blocks, and it's located inside the nucleus of a cell. If you can see my rather crude cell drawing, the nucleus it's not in the center of every type of cell. The cytoplasm, where all of the organelles and stuff inside cells are, is like a free-flowing thing. There's no fixed locations, but for illustrative purposes, we can assume that it's in the center. So DNA is a very important molecule. It's extremely complex, and without proteins, well, no life on Earth would exist. But regardless, what we want to do is we want to extract the DNA from the nucleus, and in order to do that, it's a little bit difficult because we have to break some things apart. Protein chains, everybody knows what a protein chain is. Well, I'll actually put a link in the description because I just realized it's a little complex. Anyway, the other two words we have to describe are the cell membrane and what octoploid means. Now, the cell membrane is nothing more than this, not the outer green line, but the inner one. The inner green line is the cell membrane, which is composed primarily of what is called a phospholipid bilayer. It's just the type of molecule that the cell membrane is composed of. Now, what makes the phospholipid bilayer rather interesting is that it basically acts not as a pump, but it can selectively allow types of molecules and ions to flow in and out of the cell, keeping foreign material out and other stuff that it wants inside of it. Basically, think of it as like having really, really tiny gates all along the inside of this cell membrane. Now, on plant cells, we have something else called a cell wall, which actually encompasses the rest of the cell and makes it a very rigid structure. The cell wall, if I'm not much mistaken, is composed primarily of cellulose, which is a pretty tough substance, difficult to digest, difficult to break open, but that's fine. Now finally, octoploid. Octoploid is the reason we're using strawberries instead of something like bananas. Octoploid simply means that there are 8 sets of chromosomes, rather than however many you'd find in something like a banana cell or I think white blood cells are the only one. No, red blood cells have DNA in them, or like a red blood cell, right? Strawberries are the only fruit that I'm aware of that are octoploid in nature, meaning they simply have more DNA to extract than any other type of food. Excuse me. Of course, strawberries are expensive, but I'm only going to be using one in this instance. Now, a chromosome is nothing more than bunched up DNA, I believe. Humans have 2 sets of chromosomes, XX, XY. I don't know what the names are for the chromosomes for strawberries. But the point remains that the fact because strawberries are octoploidal in nature, it means that they essentially have more DNA to give out, to the point where when you extract it, it's actually quite visible, easy to see, easy to manipulate. So hopefully my camera will be able to pick up that image. Now, on to the fun stuff, the actual procedure. The procedure for this, let me back up. The procedure for this requires very simple materials. I've already measured out a 24 milliliter saturated sodium chloride solution. You'll need about 3 milliliters of dish detergent, same detergent we used for the previous video I uploaded, the elephant toothpaste video. And an equal volume of an organic solvent. Now, here's the thing. The materials differ for DNA extraction, but the procedure, whether you're in a professional lab or doing it at home or school, is typically the same, or at least very similar. We want to break open the cell wall, we want to break open the cell membrane, we want to get to the inside of the nucleus, and dissolve everything else but the DNA, make the DNA insoluble in a solvent. Now, you can use different types of solvents for this, as long as it's... the two most common ones that you can get your hands on for a home lab would be methanol or ethanol, and in my case, I'm picking ethanol, also known as isopropyl alcohol. Incidentally, isopropyl alcohol is much better for smaller yields. It has something to do with the solubility of different types of salts in the cell. I don't know the reason off the top of my head. But for larger extractions, you typically want to use something like methanol, or even in the lab they use chloroform and benzene or something as their solvent. And they use like centrifuges to bust open the cell, but the process, even though they have fancier equipment, is generally the same. It's basically manually busting open the cell and then dissolving everything chemically and getting a solvent that the DNA is insoluble in, so it'll crash out, and we can actually extract it that way, which is what we're going to be doing. Okay, so let's get started. Oh, sorry, I forgot to mention. So the dish detergent, or the soap rather, will actually act as a solvent for the cell membrane. It'll help dissolve it, along with us being able to crush it up. And the sodium chloride solution, if I'm not mistaken, will actually help break up the protein chains holding the DNA together, releasing it into solution. That's the reason we're using these materials over here. Let's get started for real this time. So I have here one strawberry in a plastic Ziploc bag. our reagents, I guess, our materials that we need. I'm going to go ahead and measure out 24 milliliters of the saturated sodium chloride solution into a test tube. Just want 24. And 3 milliliters of dish detergent. Did I say 3 or 4? 3. For my phone, I'm going to have to get something like a tripod, or something I can just prop the phone on. 3 milliliters. Assuming this doesn't take forever. This is riveting footage, I know. I actually bought this funnel separately on eBay. 2, 3. Whoops. That's fine. Okay. Now we're actually going to combine these two into their own solution. The reason we want to use such a small amount of detergent is because Otherwise, it's just going to make a mess of the entire thing. Rinse this out so we get. So we get all of the soap. Normally I would actually just edit this out in post-production or whatever you call it, but I hate video editing software as it stands, and I don't know how to use any of it, so I apologize if this is very tedious work. It is tedious watching this. You can go ahead and fast forward past this point, or if you like hearing the sound of my voice, you can continue to listen. So we have our solution here and our strawberry. What we're going to do is we're just going to put this in a Ziploc bag or an appropriate container, and we're going to manually bust open the strawberry. Let me set my phone down. Actually, let me pause this. So I've poured the salt soap solution into the plastic Ziploc bag with the one strawberry, and we're going to go ahead and mash it up right now. I kept the strawberry in the freezer, which might have been a poor choice on my part because it's rather difficult for me to break out. Yeah, I'm going to need a hammer. Shit. Hey, look what I found. I thought putting the strawberry in the freezer would actually help break open the cell by having the water inside it expand and rupture the cell from the inside out. But we'll see if that affects yield at all. So when your parents tell you or ask you what you did for your career, you can tell them, well, I mash up strawberries for a living. That is my job. I take hammers to strawberries. I don't know what the hell I'm talking about. Nice gunk there. Oh, that reminds me. So, I know you can't pick it up on camera, but when we filter this broken up strawberry off into the beaker over there, I noticed that it actually smelled rather bad. Because I personally love the smell of strawberries, but my guess is that because we ruptured the cell membrane and released all the organelles and stuff into the atmosphere, because we released them essentially, that gave off a particularly unpleasant odor. It's not super bad, but I was doing some research into different types of not different types. I was doing some research into how to either extract or synthesize natural smells of foods, and I came across esters, which basically have various types of smells, mostly fruity. And for the next video, or at least a very close future video, what I'm going to be doing is I'm going to be attempting to synthesize an ester, methyl cinnamate specifically. I don't know if that's how you say it, cinnamate, whatever. Because it's actually supposed to be one of the main components in responsible for the smell of strawberries. I'll actually get more into detail about that in a future video. But for now, let's continue with this synthesis. Extraction, not synthesis. This part is a pain in the ass. I don't have anything like a separatory funnel. Let me put my phone over this. No, that's not bad. This way. I'm not going to make you watch the whole gravity filtration here. I'm just going to show what a few drops look like. Incidentally, if you have something like a, what do you call those, like a cheesecloth, you can use those and it's actually going to be a lot better than just a coffee filter, because it holds a lot more for starters. Maybe I should have used my glass funnel there. We'll see. You can go ahead and skip over this boring part. Okay, let me throw this out. Hopefully you can see we've got a steady drip going. This is going to take more than a few minutes, so I'm going to pause this video and come back to it once it's done. Okay, so it's been about 20-25 minutes, I think, and I managed to filter out most of the solids. Unfortunately, it seems like some fine particulates got stuck in there anyway, so I definitely should have used something like a cheesecloth instead of a coffee filter, but it's fine. So the solvent we're going to be using is alcohol. This one is 91%, and I actually chucked this in the freezer beforehand, so this is ice cold. DNA is less soluble in cold isopropyl alcohol. Here, let me reposition the camera. So, maybe I can get better lighting doing this. Yeah, a little bit. It might be difficult to see at first, but as soon as we add the alcohol, the DNA should precipitate out, if not immediately, after at least a few seconds. Let me keep the camera hovered over, so I'm adding the alcohol right now. Yep, I can immediately see some DNA precipitating. Maybe if I hold it to the light. You'll notice a layer at the top is formed. That is our insoluble in alcohol DNA. I'm actually going to try picking that up and putting it onto like a coffee filter underneath a paper towel or something. Let me go get one. I'll be right back. There we go. Luckily, DNA sort of aggregates or clumps together, making it rather simple to handle. That little white blob in the middle. So I'll try and scoop it out. There we go. Yeah. And here we have, no. Can't really see it on the paper, on the coffee filter, but here we have nice white clump of DNA. That's a lot. I mean, it doesn't look like a lot from my perspective, but from one strawberry, and the fact that we didn't get very much of it into solution, just goes to illustrate the fact that being octoploidal in nature means that they do have, in fact, strawberries do have, in fact, a lot of DNA to provide for yield. I don't know what the storage methods or anything of it are, or for it are rather, but yeah, hey, that's the building block of life. The stuff sells, the stuff that you find inside the nucleus. Well, thank you all for watching so much. I appreciate it. I really do appreciate the fact that whoever stuck with me for these long-ass videos all the way to the end, listening to me ramble on, I really, really do appreciate it. So thank you all for watching, and hopefully the next video... oh, shit. Okay, just a quick lab cleanup tip. I noticed one thing. The last time I did this lab, probably because some sugars got released, a lot of my glassware was sticky. So you definitely want to use some excessive soap and like a scrub brush or something to clean your glassware. Otherwise you're going to get like sugar stuck on the inside of your glass, and just ordinary water isn't going to rinse it out. Anyway, I'll see you guys next time. My next video might be on esters. I don't know if I promised making a different video on another topic, but we'll see.